how did the show get started? Lots of people, as Linda mentioned, my purpose in talking is because I want to share a little bit more about a world that not lots of people know about. Even myself, when I was first starting, depended a lot on my undergrads and my boss, Tara Youngborg, to support me and help me learn the ropes, because it is a different field and one that you really don't know about until you make the decision to be part of this world. Not the world, like the art world, that's that world. And it's been such a growth process for me. I feel I've grown so much in my career, in my education, and being able to interact with other people and different types of people. So yeah, I don't want to break any rules, but if y'all want to unmute and ask me questions as we go along, that's fine with me as well. I know sometimes I'll have a question and I'll forget it if I have to wait. So I will not take any offense if anybody wants to go ahead and jump in with any questions. But going into sort of how this show came to life, what went into it in the process, I really like to give credit again to my boss, because she's a very forward-thinking woman who tries to keep us on a good schedule, that's planned out really well ahead of time. Unfortunately, even she could not plan a global pandemic. So when we first started talking about what show we want to do in spring 21, she asked me, her relatively still new graduate assistant, hey, do you want to do a show? And I was like, oh, okay, sure, sounds great. I've helped with one. And that's really how it all started. And I knew as a woman of color in this field, I was like, this is an excellent opportunity for me to shine a spotlight on people that are often overlooked in the field. I meant to pull up some statistics for you all, but if you ever want to check, I highly recommend everybody go do it, because in the art world that is so encompassing, everybody knows an artist who looks like you, looks like me, looks nothing like you, looks nothing like me. Yet for whatever reason, the capital A art world is still very much dominated by a ever, I'm not going to say that, by a very small section of the world's population. It usually tends to be cisgendered, heterosexual white men. And not slamming anybody who loves that artwork, and that is fine, but the art world does encompass so much more, and it just continuously is being overlooked. And even in my time in the art, working in the gallery, I was like, yo, this is something that we can address, something that we can do. And Tara was like, heck yeah, we can, let's do it. So going into it, I knew that I wanted to do something that showcased the art and artists and artwork done by people of color. I wanted to bring in anybody who fell outside of that dominant narrative. That was a lot. I do only have a small space. The gallery is not that big. We are small, but we are mighty. And so over the course of just me thinking about it, I knew where I was headed. And then coronavirus shut us down and things got a bit crazy. But when we got the news that this coming fall, so fall 2020 now, that we would still be able to be open, that we were still going to do shows and all of that jazz, we really hit the ground running. And for me personally, everything that happened on multiple levels over this past summer, and again, throughout, at least, if not my life, everybody's lives, really, really helped me narrow down what I wanted to talk about. Because and one of the points that I like to pride myself on working in the gallery is the fact that our shows really tap into what our students and what our community are talking about, are thinking about, are being active about. And I really felt our last couple ones have been in that kind of vein. So last fall 2019, we had a show called Still Here, Art on HIV/AIDS, in which we were discussing that pandemic and how devastating it was, especially given that it was so overlooked for so long. And then, but right before coronavirus happened, or as it was breaking and we were going into lockdown, our show was focusing on mental health. It's called Neuroblooms by artist Leslie Holt. So all of that, I was like, heck yes, I'm centering Black women. I'm centering people of color. No, sorry, let me walk that back because I was very specific about the fact that it is representations of Black women because, and I know somebody might not like it, but every single issue faced by every minority in this country is exemplified in the struggle of Black women, on every level in different capacities. And I feel very strongly about that for a variety of reasons, including being a Black woman, being friends, being raised, having them around me. And I believe that very much it is a concern for everybody because until we have, man, I'm going to get quotey now, until we have justice for all, there's going to be justice for none. So I knew what I was looking for when I sat down and started checking out the local artists, checking out the artwork that I was seeing around me. Being located in the DMV, I know we're not like the art capital of the world, but I feel like we're up there, and that one is not statistically backed, so don't quote me on that one. But yeah, it's really easy. And another pride point for the gallery is that we focus on, as much as possible, bringing in local, regional, emerging and mid-career artists, again, to diversify who has that platform for themselves and their artwork. In a regular time, in before Corona, so BC, we would reach out to artists that we knew. There are lots of local artist boards, bulletins. You can put out artist calls. In the past, that is how we've done. We've also had people approach us. A mixed bag of how we decide what shows are going to happen or how the show is decided to come into the gallery. With this one, because I had the idea without having the artists first, I went out and started looking at those different boards. Again, Tara was darn near holding my hand through all of this because I was very, very much like, I don't know what I'm doing. I want to do it justice and I don't know how to do it. She was like, you got this, you know how to do this, and we will help you. And that's exactly what happened. After poking around for a while and trying really hard not to get too lost in all of the amazing artwork I was finding by all kinds of people, I decided to reach out to a couple. And this is the part where it gets a little misty for people who are not on the back end of the art world, because how did I get these artists? I shot some emails into the dark. Those cold contacts that everybody dreads when talking to like professors. Think of that times 1,000, because I didn't know these people in any capacity. I was just like, hey, I really, really like your artwork. Would you talk with me for a little while? And thankfully, people did get back to me. And then it was a matter of setting up virtual virtual gallery visits. The bummer is I was really looking forward to going to see these artists and artworks in person back when we could go outside. I was so excited for it. I thought that's what I was going to be doing over my summer and all of that jazz. As we all know, that is not what happened in the slightest. But everybody worked to make it work, essentially. And I was able to meet up with the artists, talk to them. They showed me some pieces. We were talking about their narrative behind their artwork. And it was a lot of a discussion, negotiation to see, will your artwork work with the narrative that I want to tell to those visiting my space, my gallery? And if so, great, let's do it. If not, maybe next time, essentially. But all of the artists that I spoke with and got to that stage of talking to them to visit their galleries and all of that jazz, it was really easy because their narrative, my narrative, were the same things. It wasn't difficult at all because, again, this, like I said, in the art world, it is a well-recognized problem by a lot of people about the lack of diversity, about the, if not enforcement of one narrative, definitely a rather rigid path set up for the narrative of art world. And that being said, me seeking to change that and them seeking to also change it, it just worked perfectly. And I don't say this to act like every art show, art exhibition just falls into somebody's lap. This one certainly did not fall into my lap. I was just grateful and blessed enough, essentially, to be in a position and find artists and people willing to work with me, that we were able to complement each other's visions in a very fruitful way. I'm going to take this chance to go ahead and change my background really quick. So these three pieces, sorry, I'm trying to watch my own thing. These pieces, there's three of these digital photos, and they actually have poems imprinted on the back of them, but each of them correspond with one of these headpieces done by artist Andre Terrell Jackson, who uses they, them pronouns. And they're actually amazing. First, let me recommend, how did I find Andre? They were actually on display on 14th Street. And I was going to El Centro during the summer or something when I saw their work in one of the front displayed areas. Okay, boom, I'm gonna try to navigate my body now, pretend I'm not there. But yeah, and I was like, oh wait, I can call this person, I can text this person and see if they want to be part of my gallery after they were in the real world. Not to say that the gallery is not the real world, but I'm sure all of you are very familiar with how campuses can seem very insular. And so to have somebody who was like in DC, like in a storefront on 14th Street, it's 14th Street. I was very excited to have all of that. So unfortunately, like I said, we did not have enough time to take all these quality photos. So there's only gonna be a couple. And then to this side of me is one of my favorite pieces by artist Akia Breon Brown, who is, I believe she's also a Hamiltonian fellow. And her star is currently zipping to the top right now. believe she has two or three other exhibitions that she's a part of going on at the same time that she's in ours. And she has previously had a solo show. Don't quote me on the solo show, but she's definitely had a show down in the National Museum of Brazil, I think. She's amazing. And these pieces are evocative of lots of emotions for those who may see themselves reflected in it. I know that I do, and that's part of the reason that I was determined to get her into my show. I'm sorry, our gallery show, my baby. And yeah, it's just been, and the third artist, sorry, I've spoken about Andre, I've spoken about Akia, and then the third artist is Sadia, the young lady who, if you saw in the chat, she's an amazing young woman. She just graduated December 2020, and she wrote, narrated, directed, and edited and acted in that video that you all just saw. So I definitely recommend y'all, if you did not get a chance to see it, I believe it was just dropped into the chat again. Thank you, Linda, or whoever dropped it. Much appreciated. So yeah, that's how I found the artist. That's how we ended up choosing the art pieces that were going to go into the show. What else is there to add? Is it helpful if I tell you all how we install things? Because that's my favorite part. It's fun. Sometimes we get to paint the entire gallery. One time we did it red, and it was great. It was beautiful. I really want to do it again one day, but it was so much work. And the fact that coronavirus means that myself, Tara, and the docents, so we are entirely staffed by undergraduate docents who do help with install and deinstallations. But given social distancing requirements and all of that jazz, everybody trying to be as cautious as possible, install this year has been a little bit wonky. So the fact that this show is actually very easy to install was also a benefit during, but when we're putting it together. All of these pieces, so we have different pedestals. So you'll see them here and here. The things that the headpieces are on are pedestals. Those are easy enough to move. And then Andre actually provided the stands for the headpieces as well. So that was an easy peasy install. We have a couple of different movable walls throughout the gallery. I'm gonna go ahead and pull this image back up. So the wall that this image is on is actually a movable wall. We slide it, it's stupid heavy. And we always, for our undergraduates, we do require that they have at least two people, but I'm stubborn, and I had a deadline and goals to finish, to complete. So anywho, we make it all happen. We move the walls around, and then our back wall, here's another image. Okay, I'm gonna like shrink here, and you'll be able to see like our back wall goes that way. It's a pretty long wall, and this is where all of the Kia's pieces are brought up. You can see another one that was different from the original one that was up. And then you can see just a little bit of this piece in the middle. I will never say that I have one favorite piece out of all of my artists, but I will say that I think that by IKEA, this one in the center is actually my favorite piece. And I recommend, if y'all really want to take a look, check our website on Monday to see if we're open again and come by if possible. If not, we are working on being able to make this exhibition visible and accessible in some way online, even if it is just pictures. Yeah, so this was a very simple install. I won't go into the details of hanging things so that there's proper spacing or proper height, but let me let you know that there's a lot of math. And I'm a history major. I hate math. I really struggle with it. I don't think I've taken a math class since 2015, which is actually impressive. I definitely finessed the undergraduate system to do that. But yeah, it actually does involve a little bit. And I will say that that is the only time that I'm willing to do more math than necessary, because you always want to measure 2, 3, 17 times and then make as few holes in the wall as possible. Yeah, I think that's, oof, I might actually be ending a little bit early. So if you have questions, but yeah, this show really has been a passion project in addition to being, my real full-time job. Um, and I'm just really glad it makes me feel as if, in my own way, at least, I'm doing something by using the platform that I have to allow other voices to be heard. As Linda mentioned, that's something that's very important to me, because in our own ways, even if it may not even be something that I personally agree with, but having that diversity in conversation and dialogue is the best way for everybody to at least reach better understanding. And it's not until we start trying to understand each other, start seeing where other people are coming from, where we find those commonalities that will start fixing a lot of the issues that all of us are living with.
So as Linda said, I'm a PhD candidate in the MEAS program, and I am centered at the Chesapeake Biological Lab in Solomons, Maryland, which is part of the University of Maryland Center for Environmental Science. So we're kind of this weird thing where we're our own institution, but we matriculate through College Park. And so we do have College Park MEAS students also. And so that's our program. And then we're just kind of at this different institution. And so today I'm going to talk about part of my dissertation. I've become, as several people on this call know, very partial to clams. So I'm going to tell you a story of some clams in the Pacific Arctic. So just as an introduction to the Arctic, it's actually kind of unique in that it's an ocean surrounded by land, as you can see in the map. So those of you in the DMV area, you could almost maybe look at this and think of it kind of as the Chesapeake Bay, where you have Maryland and Virginia on either side of stuff. There are 8 nations, which make up the Arctic Council, and they're highlighted in that map. So Canada, the United States, Russia, Finland, Sweden, Norway, Iceland, and Greenland, or Denmark. And then there's what's called 6 permanent participants in this Arctic Council. And this is the indigenous groups that live in the area. And they're all listed there, and which countries their land is and where they're from and where they live. So that's just kind of a brief introduction of where we're going to be. When I say Pacific Arctic, I'm talking about this side over here. So the United States and Russia, as well as Canada. And I focus in the United States and Alaskan waters. So why the Arctic? It's really hard a lot of times for people to get jazzed about something that seems so far away, but it's one system and there's lots of connections, both physical and cultural, across regions. I'm actually really glad the other student talk tonight, Brianna, was an arts and humanities one, because I think it's really fun when we can mix kind of these two worlds. And so we have to kind of protect it and think of it as this system as a whole. And the Arctic is rapidly changing. So this graph here is an example looking at surface air temperature, which I won't talk about air temperature again in this talk. Everything's going to be marine, but it's increasing at almost twice the speed of the global average. So in that graph from 1900 to 2020, the blue line, that 60 to 90 degrees north, is the Arctic, and the global average is the red line. And you can see that that blue line since about 2000 really has skyrocketed above that red line. And one example in the US Arctic that I'm going to come back to that's really important to the rest of the story is that there's been significant warming of bottom water temperatures in the northern Bering Sea, which I'll show you a map in a minute. And we're talking about 1.5 degrees Celsius higher than what's previously been recorded in the region. And so because this is interdisciplinary and we're trying to learn about each other's stuff, I tried to kind of phrase and set up my talk and my questions using Charles Dickens' A Tale of Two Cities. So I've sort of rewritten, which is why it says Dickens-ish, 1859, kind of rewritten that famous introduction paragraph of, was it the best of times or was it the worst of times? And so I'm going to use this to kind of set the stage for the story in the Pacific Arctic and try to tie in some English and humanities into this science talk. So starting off, it was still the age of limited data, but it was finally the age of time series analysis. So what does this mean? The Arctic is what we would consider a, quote, relatively new area of exploration. This is a list you can see going back to about 1826 of perceived firsts. So the handsome gentleman in the left there was the first to explore the Alaskan coast from Point Barrow, so a northern point in Alaska, to the Bering Strait. And then as we kind of move through the century and into the mid-1900s, you can see there was a ship frozen into the pack ice for the first time, hoping to get to the North Pole. Robert Peary and another group, although maybe this is contested, there's other people that claim this, were the first to reach the geographic North Pole. And Roald Amundsen or Richard Byrd claimed to have the first air expedition to the North Pole. And so the point of this is to kind of show that, 1926 is a year we can kind of wrap our heads around. This is post-World War I, kind of just before World War II. So it's a very new area of data collection. And so what that means is that, like I said, there's limited long-term data available, and for a variety of reasons. So sampling in the winter is very challenging or not feasible, although possible that that's becoming more open with changes to sea ice. And so a lot of our data is only collected in the summer and early fall. And while, a lot of people that are working with time series get, they have 150, 200, 300 years worth of data, I get really excited with my data set to have 25 consecutive years of data. But that is changing. So the age of time series analysis, I work with my advisors within this project called the Distributed Biological Observatory. And the goal of this project, as it says there, is linking physics and biology. And the sites that we go to are these ones highlighted in the red boxes on the map on the screen. And so this was first proposed in 2009, and it had its first pilot field year in 2010. And since then, there are multiple national and international cruises, both seasonally and yearly, so kind of that spring, summer, fall time. And we have up to what we call 8 transect lines. And we look at this as what we call a change detection array. So looking for changes in the ecosystem in the form of productivity or rates of change. And it's kind of along this latitudinal gradient. So going from the south up to the northern parts of Alaska and into the Canadian Arctic. And we're measuring in these red boxes what we're calling regional biological hotspots. So it's an area where there's a lot of activity or a lot of animals. And in this talk, it says I'm going to focus on region 1 and 3, but it's lying. I'm only going to focus on region 1, which is in the northern Bering Sea. And so to kind of introduce you to our cast of characters, I don't have people from London or Paris like Charles Dickens, but what I do have are clams. And so there's 3 species that we're going to talk about today. And when we collect these samples, this middle picture is what it looks like when we pull a sample up and rinse all the mud away. This is a station that gets me really excited because you can see all of the clams. Other stations look different, but I'm very partial to clams. And so we're going to talk about Macoma calcarea, which will be the white one. And apologies in advance, there's not easy common names. So we have the white Macoma calcarea, and then these two brown clams, which are Nuculana species. And before I dive in and tell you about the clams, a lot of people want to know why we care about clams. So clams are a really important food resource in the region, and things like walruses and spectacled eiders, which is a diving seabird that's endangered, feed on these animals. And these animals have cultural and economic importance to native communities in Alaska, like Uktiavik or Barrow, Alaska, up in the north and along the coast of the state. So how we collect the data, usually it's more exciting. I love being in the field, and so I thought I'd share what that kind of looks like. So we collect a lot of the water data that I'm going to talk about using this thing on the left called a CTD rosette. So the instrument on the bottom measures conductivity or salinity, the saltiness of the water, temperature, and then depth. And then each of these bottles, you see the top is kind of cocked open. What we can do with a computer program is send that down and close those bottles at different depths in the water, capturing the different chemistry and physical parameters and biological parameters of what might be living in the water column in different areas using those bottles. And then the center photo is a Van Veen grab. This is what we use to get all of the critters in the mud. So this is one coming up close. So that's filled with mud and animals and all kinds of exciting things. And then we stand at these sieve boxes that have a screen in the bottom and spray away the mud, and then package up and take all the critters home for later analysis. So one of the biggest drivers in this system is sea ice. So we're seeing earlier sea ice retreat in the spring. So it was the spring of earlier sea ice retreat. And we're seeing a later sea ice formation in the winter. So it was the winter of later sea ice formation. And so this is kind of a double whammy in the system where you're losing it sooner and it's coming back later. This graph from the National Snow and Ice Data Center shows the ice extent from September of 2020, so just a few months ago. These are actually kept up to date, like daily and monthly. I just happened to pull a snapshot of September, which is the lowest ice extent during the year. And this pink line is the median from 1981 to 2010. So from 1981 to 2010, the median ice edge would have been this far, and you can see how far back that is in 2020. And so this is looking at annual sea ice persistence. And so all that means is the number of days per year that sea ice is present. And so thinking about that first region right here, I hope you can see my mouse, I'm moving it around. You can see the pink colors mean there's more days with ice. Darker purple and black colors means there's less days with ice. So even as early as 2013, you can see as you track it through 2013, 2014, 2015, up to 2018, that box in the south that we're focusing on is having less and less sea ice, number of days with sea ice as we move forward in time. And then if we think about it kind of as concentration and look at it on a yearly scale, I want to draw your attention to the red line and the orange line. So the red line is 2018. The orange line is 2019, and then that blue line is that 1981 to 2010 mean. And so the mean, you have sea ice disappearing in June and coming back in November. If you look at the red line, it's gone by March. So that's 3 months sooner than it usually is. If you look at the orange line, it disappeared kind of mid-March, early April, came back, and then was gone before May or right around May. And then in the red line, the ice didn't come back until kind of December, and the orange line doesn't even reappear until January over here. So this is that dual story of it's leaving sooner in the season and coming back later in the season. But what does that mean? So I told you that bottom water temperatures in the area were changing. So this is the map in the corner here is to orient you to what these stations mean. So this is St. Lawrence Island, and so we're in the northern Bering Sea. And we're going to move from slip 1 in the south up to slip 4 in the north. So going up this graph, you're moving more northerly. And then along the x-axis here is the year, and we're going from 2000 to 2019. And what's really jarring about this image is you see this bullseye of warm colors. So this area has a pocket of cold water, which will come into play a little bit later in the story. And it's -2 or -1.5. These are negative degrees Celsius water, so they're below 32 Fahrenheit. But in 2018 and 2019, we had this warm water. It's up into the +2 and +1.5. So there's some really major changes going on in the physical environment of this ecosystem. So we talked about ice and temperature. The other really big driver, what we call driver in this ecosystem, is thinking about food availability. So we all, are happy when we can go and get a cheeseburger, and might be a little bit sadder if we're eating salads all day, every day. And so in Charles Dickens' language, it was the epoch of new fall blooms. So the potential for blooms later in the season, blooms of food, and it was the epoch of changing spring blooms. And I'm going to talk about this diagram on the right-hand side in just a second. So this is a conceptual diagram of what this system might look like. The clams that I am really interested in live in this realm, the benthic sea floor. And then you have animals that are in the pelagic ecosystem, which means they're living in the water column and feeding on things in the water. And then you have your ice and your ice pulling back. And we get this thing called an ice edge bloom. And what happens is a lot of this, there's a mismatch in timing of when the animals in the water column are feeding and when this bloom occurs. And so a lot of that bloom of food material falls to the bottom, which feeds a really rich benthic community. So the clams, the worms, small crustaceans, and we get what's called a benthic dominated system. And so you have this algae that falls to the benthos and feeds things like the walrus and the diving ducks that I showed you earlier. And then kind of this smaller pool of zooplankton or the things that live in the water. But as those warming seawater temperatures come in, what that could do is potentially increase zooplankton, the little critters in the waters, their growth, their abundance, so how many there are, and their grazing or their eating. And so if that increases and starts matching timing of blooms better, there might be less food that is deposited and available to the bottom. And we might end up with this kind of shift to a different structured ecosystem with phytoplankton feeding zooplankton and feeding things like fish that live in the water column and different seabirds and whales, and get kind of this shrinking of the benthos. So what does that all mean for our cast of characters? So it was the season of shifting hotspots. So remember, those hotspots are those areas where there's lots of stuff going on and lots of animals present, and the season of predation patterns responding. So this is where our birds are going to come back into play. So to reorient you to our distributed biological map and our northern Bering Sea region, I'm going to focus in on that region and zoom in. So this is looking at what we call biomass, so kind of the weight and availability of material for eating. And we're going to focus in on those orange bars. And so again, we're kind of thinking about what we would pull up in the grab. And slip 1, 2, and 3 are kind of in our southern part of our box, and they're declining. So this means there's less available benthic biomass or weight available for food. And in our 2 northern sites, we kind of see a no trend yet. But overall in this area, the benthic biomass is declining. Not only is it declining, it's also shifting what's dominant. The purple bars are our clams, and you can see kind of here's our 3 southern stations again, and blue is starting to creep in a lot more than the purple, kind of post 2009. And those blue bars represent polychaetes or worms. And you can see in the north, our clams are still pretty dominant. This only goes to 2015. I'm actually in the process of sorting. I just finished sorting samples from 2016 through 2019. So that data will be available soon. But we're seeing a shift not only in biomass, but also composition. And so this is where I really get interested in, okay, of those clams, who's present and how might they be changing. So these blue squares represent our nuculana, so little brown clams. And you can see from 1985 to about 2010, they've been declining in this area. And the purple star is our white clam, and there's only one dot there. And so that's part of my dissertation is filling in more purple stars. So what does the data look like? So I look at two different kind of metrics or measurements of the clams in the area. I look at both abundance and biomass. So abundance is simply a number of clams per meter squared. And so dashed lines in the next set of graphs are going to be the nuculana, the little brown clams, and solid lines are going to be the macomas. And so now we're looking at even a story between shifting species of clams. And so you can see nuculana has been declining, but then in 2012, we started to get an increase again. And so that might look really exciting, but we need the biomass to tell the full picture. But we have kind of a story of macomas kind of coming in, declining, but maybe ramping back up, right? This is almost 350 individuals per meter squared. And then in the north, kind of the same thing. We see this decline of nuculana, but this increase in macoma. But the biomass kind of tells a really different and important part of the story. So you can see nuculana just completely falls off. So we had that increase in 2012 in abundance, but the biomass isn't there. And so what that tells us is that there's a lot of clams, but they're really small. So if you're hungry and you're spending energy to get food like these birds might be, if you're having to sit there and pick tiny pomegranate seeds or something that's really small and try to fill yourself up with that, it's going to take a lot of time and it's not going to be really energy efficient. Whereas if you can have something more like the size of a pancake, it's going to be more filling and more fulfilling with the energy that you expend. So the macomas, the biomass is increasing both in the north and it's starting to increase again in the south. You can see it kind of dipped down. And so the food source here is changing. And there might be a lot of clams, but they might not be the most calorically or nutritionally efficient for these predators. And so in thinking about the predators, and I tried to, alliterative bivalve birds in the Bering Sea. This is our map of our DBO sites again. And then this one shows the range of the spectacled eider in Alaska. And this is the overwintering ground for the population of the spectacled eider. So they come here and they feed. They are endangered and they are feeding on these clams. And so there is a size class of these clams that is the most calorically beneficial. So understanding that and how their prey is changing can really help us understand the ecosystem and the trickle up effects both to top predators as well as the physical environment and kind of the bottom up controls. And so there's an importance too for this that I'm going to talk about in a second for the local communities. But remember I told you there's this cold water there and it's we might be kind of shifting to maybe new predators or a new pelagic or water column ecosystem. So this is a slide of NOAA trawl survey data looking at Pacific cod and pollock in the Bering Sea. This pink is that cold water that I showed you in the other colorful graph, and all the blue dots represent the fish. So the darker the blue, the more kilograms per hectare, I think is the measurement. So the more fish essentially. And you can see they ride along the edges of this cold pool. So they're not going into the cold water. So this cold water kind of acts as a refuge to keep, for the benthic animals to keep the fish out. But you can see in 2017, it shrunk, the cold pool shrunk, and they still hung around the edges. But in 2018 and 2019, when it was non-existent or barely there, you can see the fish moved into that region. So this is indicative that these temperatures are changing and things are starting to shift and come in, and we're seeing animals further south kind of move into the region. And I'm really hoping this works. This is the most interactive part. This is actual footage of the bottom in this area. Lee Cooper, who's on the line and one of my committee members, sends down a camera and we go out in the field every year. And these green dots are 10 centimeters apart. And what you're gonna see is a really big fish. To us, that's a really big fish. That's what we called it for a long time. We found out it was a pollock. or was told it was a Pollock. And I'll just play it again because I like this slide. But we haven't seen one on the camera before, and this was, you know, an example of our data matching some of the trawl survey when we put this camera down. So moving forward, kind of under unprecedented change, which is a phrase I'm sure we're all actually tired of hearing under COVID, is unprecedented, but the ecosystem in the Pacific Arctic is starting to potentially become unprecedented change. So we have these warm bottom waters in 2018 and 2019, which could be opening up to new fish predators with that physical barrier gone. And we've got this shifting kind of benthic community from worms to clams to worms, sorry. So what does this mean moving forward for the bivalve population in the ecosystem in the future? And that's where I'm going to do a really quick snapshot of the last part of my dissertation, which is trying to understand the ecosystem as a whole. And to do this, we send down another piece of an equipment called a HAPS core. So this goes down and comes back up with essentially undisturbed bottom. And so this allows us to look at the most pristine form of the bottom that we could try and look at. And I set up an experiment with this looking at two different temperatures, so kind of the normal temperature of the region and increased temperatures that are predicted potentially for the area. And then like I said, food is another big driver, so we fed half of them and did not feed the other half within each temperature. And what we were measuring was oxygen respiration and nutrients, and so looking at carbon cycling in the region. And this is a picture of my advisor, Jackie, and I in a cold room with all of our cores. We spent a lot of quality time. It's a little weird to walk into a room that's set to 1 degree when you're in the Arctic, so you're just kind of constantly surrounded by cold. And then I also do that with individuals. So this last picture on the right-hand side is looking at individual clam respiration. So I want to know what is their contribution to the overall system, because I've sat here for the last like 10, 15 minutes telling you they're really important. And so if they're that important, I need to understand what their role is in the full system. And so just an example of that and what a measurement might look like if we take one of our northern sites at 4 degrees, so a warm scenario. A core might have a measurement of oxygen, millimoles of oxygen per meter squared per day of about 14. And these are actual data points that we've collected. And one individual, so this is a third clam that I snuck in there called E. tenuis, might have something around 3.5 millimoles of oxygen per meter squared per day. So that could potentially make up almost 25% of the total. So these clams are really important kind of movers and shakers in the ecosystem. And so kind of bringing it all back together and drawing hopefully a complete photo of this system and the importance of clams. So I think about the past and I look at change over a 15, 20-year time period, thinking about an individual and how the drivers for that individual are its availability of food, the physical parameters of ice, and more consequently, what the temperature on the bottom and what they're living in. And then thinking about it kind of in the present on a population level. So how are these clams interacting with one another? How are they interacting with other species like worms? And what physical drivers are changing that, which as you can see are the same physical drivers usually that are driving individual populations, and how that might affect their predators, so the birds or the fish that might be feeding on the worms. And then thinking about it into the future at the ecosystem and community scale, using these sediment chambers and looking at individual contributions to the whole system. And again, how that might affect the predators, and to tie it all kind of back together to these local communities in Alaska that really rely on the resources. I'm working with a woman now on a conference section actually that is presenting a poster called "The Ocean is Our Grocery Store." So these communities really are an integral part of this ecosystem and really rely on this system. And so with that, I could talk about clams for hours. My artistic expression, I still draw the sun like Brianna does, but as some of you know on the call, I express myself through my clothing. I actually have clam leggings and wear them as much. They're bright colors. Black is actually not my norm, but just to say thank you to all the people that have really been involved in this, my committee, all the lab members, the boat, the funding agencies, etc.